Hi, folks. I'm Dr. Kevin Hagedorn, and our company is Life Magnetics Incorporated, and we do RNA transportation and storage. RNA transportation and storage is actually quite a large market in biotechnology. Do you remember how the vaccines need to be cold stored? And it was in the news every couple of weeks that there weren't enough freezers when the vaccines were first starting to get rolled out, and they had to do the injections. It couldn't just be a pill. And the COVID-19 testing, at-home PCR testing wasn't available. Well, all of those have to do with the fact that you can't easily transport and store RNA. By addressing this problem, it's actually tapping into, by some estimates, nearly $1 trillion market. RNA transportation and storage is important for a range of opportunities in life sciences. In therapeutics, Moderna said that they developed the COVID-19 vaccine in just 1 or 2 days. The rest of the time was basically clinical validation. It's very easy to develop a sequence coding for a specific protein or biologic, which can be translated in the body. The problem is that RNA is generally fragile, so it's difficult to get it in a stable way into the body, thus the need for the -80 freezers. But if you've got an easy way to deliver RNA in pill form, this could potentially be a $320 billion market according to some sources, or up to a $700 billion market according to other sources. And we've been working on this in collaboration with Asperovax. Another of our applications, though, with an earlier market opportunity is environmental testing. In farm testing in particular, bird flu in the fall cost the UK government nearly $2 billion from turkeys that they had to put down. In farm populations, pathogens can spread like wildfire through the environment, but testing each individual animal is not economically viable. Environmental testing allows us to collect samples in the field and basically screen the entire population simultaneously. This allows us to find rare diseases in human populations or identify outbreaks on farm animals at a more reasonable rate for farmers. Or in diagnostic testing, we'd like to do more at-home testing, like urine testing for kidney transplant rejection or prostate cancer. Exact Sciences did very well with their Cologuard kit. That's the poop in the box you see ads for. More at-home testing that allows people to avoid scheduling doctor's appointments has been a very successful model. Being able to collect RNA in the field and transport it back to the lab effectively would open up new opportunities. Or in agricultural testing, for example, RNA-based pesticides are predicted to eventually replace small molecule pesticides. So in rural areas, cancer rates are 20 or 30% higher. That's because the pesticides we use sort of linger in the environment. RNA degrades relatively quickly. So if you have a way to target a field, eliminate the pests, but then not have that runoff persist in the environment, it's predicted that many governments would make small molecule pesticides illegal, and RNA transportation would enable that. Our solution to this problem is a carbon semiconductor that binds to and stabilizes nucleic acids when calcium is present. And we sell this as a kit primarily right now for wastewater-based epidemiology and for lab-based testing, with applications developing in diagnostics and therapeutics. One of our more successful areas has been in environmental testing, where wastewater-based epidemiology is a growing field in life sciences. This is really a blue ocean market. Wastewater-based epidemiology really only became a thing with COVID-19 testing in human populations, but we've identified that farm-based wastewater epidemiology might actually be a larger market. And we've really got a significant advantage in this area. We've cut the process time down from 2 days to just 2 hours. We've eliminated the volume requirements. We've increased sensitivity by 100-fold. And we've reduced the cost, importantly, down from about $850 per test to about $150 per test, mostly by automating and removing labor from the testing. This is currently in a pilot with Michigan Health and Human Services and with Central Star of Michigan that tests 5 million cows in the tri-state area, basically. This is one of our data sets from our wastewater-based epidemiology testing, where we're detecting COVID-19 and its variants in cities around Michigan. This particular type of testing is important for farms because there's a number of pathogens that infect cows, chickens, and pigs that are difficult to detect through routine testing. For example, one pathogen is bovine leukemia virus. Bovine leukemia virus is mostly symptom-free until cows start developing leukemia between 4 and 8 years of age. So you don't want this spreading through your cow population. In large farms, it can cost millions of dollars, but it's difficult to detect, and the blood tests used to detect it are relatively expensive to do individually. So through this wastewater testing, we were able to detect COVID-19 and its Delta, parental, and epsilon variants. This same type of testing could be used to simultaneously screen for bovine leukemia virus, bovine tuberculosis, Asorius infections that cause mastitis, and so enable farmers to address these problems before they grow out of control in these closely confined areas. And in at-home urine testing for kidney transplant rejection, we're able to show that the RNA once bound to our carbon beads is stable for 30 days plus, which again, the standard time that RNA is viable is about 7 hours. So this is a dramatic improvement over the current state of the art. We also have quite a bit of data in drug delivery. So the mechanism we use, the carbon beads bind RNA when calcium is present. The concentration of calcium is normally 12,000 times higher outside of versus inside of cells. So what happens is after the particles are taken up by the cells and escape the endosome, the calcium concentration is depleted and the RNA is released, which then translates inside of the cells to make the therapeutic product. We've been able to show that in cell cultures, this is less toxic and just as effective as the industry standard lipofectamine. Compared to competitors, most of the competitors working in the space use the calcium concentration gradient in order to deliver the therapeutic payload. So for example, 3.7 billion was invested in ORNA therapeutics, 50 million in Pfizer, and 23 million in Agrospheres. This was just in the last 6 months. And most of these companies leverage the calcium concentration gradient in order to deliver the therapeutic payload. However, how these companies do it is they load the RNA inside of a lipid crystal nanoparticle or lipid nanoparticle, and when the calcium is depleted, the lipid breaks apart. But this changes the chemical environment inside of the cell. And since there's actually more of the lipid material than there is of therapeutic payload, the concentration gradient of lipids and the crystal can be significant inside of the cell, and this leads to higher toxicity. The mechanism that we use is interpenetration of the electron wave functions through pi-pi stacking, which is a strictly quantum mechanical mechanism, but it allows us to use a solid material which otherwise doesn't dissolve inside of the cells. And by doing this, we lower the toxicity. We started this company actually quite a while ago in 2015, but at the time, people didn't really realize the value of RNA transportation and storage. That mostly changed with COVID-19. Now, virtually everybody is familiar with why this is important. In fact, in our first pitches, we weren't even allowed to say RNA. Our ribonucleic acid was considered to be too technical for the audiences that review these pitches. But now this is considered elementary school material. So the two people leading this are myself and Rishab Kalla. Rishab is a PhD from the University of Michigan who worked in Rich Miller's lab studying aging research, and I'm a material scientist. And that is our presentation. Thank you very much.